in Boston. So with that, I'm looking forward to your talk. Thanks.
Great. Thanks so much for the introduction. Thanks so much for the warm welcome. Excited to see familiar faces and then also a variety of new faces. These are my disclosures. All right, just to start off a little bit light. So this is, we're talking a little bit about COVID. This feels like kind of is a different era in human history, all pre-COVID. This was Disney World, my first trip to Disney World. I grew up in Los Angeles, where Disneyland was around the corner. This is me here with my small twins. It was about a year and a half ago. And then 2 months later, this was basically all of us, right? This is, my wife is Pulm Critical Care for those who don't know her. But, we were all on the front lines dealing with a disease that none of us knew anything about. And there was, rapid efforts in trying to understand the bases to, really combat this new disease. But not too long ago, there were, other diseases that are quite common today that we really didn't know much about, namely cardiovascular diseases. This was a, very important anecdote into the heavy investment from U.S. government to study cardiovascular disease. This is the death of our president, Franklin D. Roosevelt, one of the few U.S. presidents who died in office, as you all know. And he ultimately succumbed to a hemorrhagic stroke, but he was under the care of the best that modern medicine had to offer at the time. And as you can see, documented blood pressures leading up to his death, ultimately to the 300s, would make all of us cringe. And this basically goes into the reduced understanding at the time of the key drivers of cardiovascular disease, and then in turn a reduced insight in trying to understand the strategies to prevent cardiovascular disease. And as you can see, the report in the New York Times from his physician said, came out of clear sky, despite clear blue sky, despite these documentation. But I would say there was heterogeneity among his physicians. Some, were concerned about the blood pressure and some less so. Okay, so that anecdote, and then in general, the epidemiology of the rising prevalence of cardiovascular disease and increasingly premature cardiovascular disease for atherosclerotic cardiovascular disease, which was not as common, in the prior decade. That was an important impetus for the U.S. government to form an actual body to start to fund research to try to understand the root causes. And this ultimately led to the establishment of the National Heart Institute by President Truman, that ultimately, developed into the NHLBI. Okay. And this concept of trying to find the root causes and then address those root causes in patients is not a novel concept. Really, since President Obama announced the Precision Medicine Initiative in 2015 at his State of the Union address, the name has become quite widespread, but the general notion of prevention and treatment strategies that take individual variability into account is not a new concept in medicine or in cardiovascular disease. For at least 60, 70 years, there was an appreciation of at least a small number of risk factors that correlate with cardiovascular disease risk. And, over the subsequent years and decades, additional evidence linking a causal relationship between these risk factors and cardiovascular disease. So today, when we see abnormalities in these factors, we often treat, either with non-pharmacologic strategies or pharmacologic strategies or behavioral management. And then to some degree, there is also some notion of, using these factors together to estimate risk as well. So that concept is not new. I think what's important when, the NIH and others were thinking about what precision medicine should be, is now the new advances in the abilities to better capture inter-individual variability beyond those 5 risk factors that we described previously. And this is the advent of large biological databases, emblematic is now that the human genome sequence was fully available, obviously in 2015, and then additional omics, proteomics, metabolomics, the downstream effectors of genetic variation, and then also, reflecting much of the environmental component, and then even using things like mobile health technology, and importantly, the advent of the tools to actually be able to analyze these. This was, the promise of precision medicine. I think as many of us think about it today, just because the roots of precision medicine have been in genomics and thinking about inter-individual variability as it relates to the germline genome, I think many of us in our mind think about this as sort of the anchoring concept. Things like polygenic risk scores, pharmacogenomics, Mendelian mutations, and thinking about how those would influence the care for a specific patient. And we do a lot of that work, but I'm not going to talk about that aspect today. And that's because, as one thinks about what are the important determinants of atherosclerotic cardiovascular disease, age is the most important determinant of estimated risk. This is a very nice modeling exercise from colleagues at Northwestern, where they took the standard 10-year pooled cohort risk equation, 10-year risk for atherosclerotic cardiovascular disease, separated into non-Hispanic white men and African-American men, and then just said for single risk factors, how does the risk change, and then binned it by different decades. And you find, individuals who are older are always at high risk. And that is a clinical challenge, right? So one, it's hard to differentiate risk at older individuals. And two, what are the age-related factors that are driving such substantial magnifications in risk? And this is obviously not an issue just in the US. If you look at the epidemiology worldwide, as there's been a shift in cause of premature mortality from communicable diseases to non-communicable diseases, and largely due to complications of ischemic heart disease. That represents the sort of bluer bin in the middle. These represent the plurality of premature deaths worldwide. And again, the x-axis here is age. Most of these are happening when individuals are older. And there's some component of obviously the germline genetic exposures, but again, lots that is poorly understood of the age-related factors. So important cells that are involved in the development of atherosclerosis and atherosclerotic events are circulating white blood cells. So, starting with hematopoietic stem cells and a variety of inflammatory cells. They interact with many of the well-established risk factors that I talked about, like circulating atherogenic lipoproteins. They together will influence the development of plaque, a variety of morphologic features of plaque, and then ultimately predisposing to events. But they're key and necessary features. And most of these that are particularly implicated are myeloid cells. These are the cells that are circulating monocytes, become resident foam macrophages. They take up cholesterol. They interact with a variety of inflammatory cytokines. You see a selection right here, like IL-1 beta, P-selectins, and others, to promote plaque and then to promote events as a necessary component of this sort of milieu. Importantly, the properties of hematopoietic cells change as they age. Like a handful of other stem cells, these stem cells are continuously regenerating. And as we age, the ability to handle errors that come with DNA replication from cellular divisions also decreases. So first, as we age, there's more of a shifting towards myeloid progenitors of these hematopoietic stem cells. And mutations occur, they occur stochastically, they occur also as errors in DNA replication. And in general, the machinery for younger hematopoietic stem cells can rectify those errors. But older cells have a harder time and may actually lead to propagation of those errors. So there is this, potentially new concept of precision medicine, of not thinking about the genetics as a static concept, but as more as a dynamic concept, where each cell has a baseline template for germline germline genetics. But as we age, through a variety of factors, to some degree heritable factors, but largely environmental factors, the DNA sequences of each of those cells can change. Okay, so what is the expected rate of change? It's been estimated that for every decade, there is one coding mutation that occurs in a hematopoietic stem cell. And most of those will have no functional consequence. Some of them will lead to that stem cell clone dying off, and every once in a while that could promote vitality. If you look at individuals who have leukemias and sequencer leukemias, not a point that we take for granted, but it's many common genes that are recurrently implicated with pathogenic mutations in those leukemias, like DNMT3A, TET2, FLT3. Okay, and if you take individuals who have leukemias and then you sequence their leukemia cells and then you sequence their non-leukemia cells, actually their non-leukemia cells often will also have similar leukemia mutations, indicating that at some point, some earlier point, the mutation arises, and then there are some additional changes that lead to the transformation of that leukemia. Again, just taking a step back, these are, again, the hematopoietic stem cells that are important also for atherosclerosis. So the concept of, it was not really clear that, the time frame of when the mutations occurred and then ultimate transformation to leukemia. Is there a state where these mutations occur? There's some relative expansion, but then, a limbo where they're not quite in leukemia. And if it's a long window, then maybe it could be a common phenomenon. So really the first to appreciate this was a group led by Gary Gilliland, where, they looked at X inactivation in blood cells in women. So as people recall, we have two X chromosomes, but in somatic tissues, only one X chromosome is expressed. That process of selecting which X chromosome is relatively random, process called X inactivation. And there is an assay in the blood that you can test how random that X inactivation is, and it should be relatively random. So in this study, they did X inactivation studies, but they looked at women at a variety of different ages. And they found that among older women, it was more common to see skewing of the X inactivation, suggesting that for some of these women, there was a subset of cells that were selected for vitality and selection, essentially, in clonal proliferation. But again, these were women who were healthy and normal CBCs, had no blood cancer. Fast forward to a couple decades later, really with advances in genetic sequencing, some of those women were sequenced and they were found to have pathogenic leukemia-causing mutations, but again, did not have leukemias. These were expanded. So again, some earlier state, some earlier condition. This led to the term clonal hematopoiesis of indeterminate potential, or CHIP, which is a terrible acronym because every subfield in medicine, unfortunately, has a CHIP, but this is what we have. But it belies the notion that there is this sort of, this state that occurs, but not everybody will go on to malignancy. There are some necessary changes that need to go from CHIP to dysplasia and then ultimately neoplasia. So again, still not totally sure how common a phenomenon this is in the population and whether it's really only a cancer implication. What allowed us to start to look at this was that, again, advances in genomic sequencing technology, namely whole exome sequencing, where for a given individual, we can enumerate for for their coding sequences, the individual base pairs. And we largely use peripheral blood, white blood cells whenever we draw blood and use phlebotomy. It comes from these inflammatory cells. So instead of looking at germline genetic variation, we can use alternative approaches to look at acquired mutations in those blood cells. And it has some important technical challenges because typically when people are looking at genetic variation in tumors, they're comparing tumor tissue versus normal for a given patient to help filter out the germline variants. And that normal is typically blood. So when looking for CHIP mutations in the blood, that's basically the only tissue that we have. So it uses a variety of reference data, internal controls, a fair amount of manual curation to ultimately say that this is a mutation that is acquired, not germline and not artifact. Importantly, these individuals don't have any abnormalities on their CBC, no cytopenia, no dysplasia or neoplasia. They don't have diagnostic criteria of other non-malignant clonal phenomena. And the somatic mutation needs to have shown some evidence of selection and expansion. So this VAF, or variant allele fraction, of at least 2% in the blood means that at least 2% of the DNA in the blood harbors the mutation. So if it's a heterozygous, that's about 4% of the cells. So again, there's been clonal proliferation. So what allowed the initial prevalence estimates were ongoing whole exome sequencing analyses to look at the heritable basis of a variety of other conditions. There was this one study looking at neuropsychiatric disease. This study was local as a partnership with David Altshuler when he was here looking at type 2 diabetes exomes. This was looking at the heritable basis of solid cancers. And they converged on this notion that CHIP was actually reasonably common. It's correlated with aging, such that individuals who are in their 70s, otherwise healthy, it was about 10% of individuals. Again, expanded cancer-causing mutations. If you look at many of the risk factors for CHIP, actually there is a lot of cardiovascular disease risk factors that have been correlated with CHIP. Again, age, obviously. There's some interesting differences by ancestry that have been recapitulated now, actually more common in individuals of European ancestry, less so in Hispanic ancestry. We're trying to understand why that might be the case. Type 2 diabetes is a consistent risk factor, at least in these cross-sectional analyses, and folks are looking to see if type 2 diabetes could be a consequence. There's some interesting sex differences that we'll get into as well. So most of the mutations are mutations in genes that have been previously described to cause cancer, namely DNMT3A, TET2. These are epigenetic regulators. ASXL1 is a cell cycle regulator, CJAK2, and things like that. So not surprisingly, these individuals are at a very high risk for hematologic malignancy, 11 to 13-fold, so relatively high risk. Those who have larger clones have a greater risk. But again, the background rate of blood cancer is still pretty rare in the general population, so the absolute risk difference is still reasonably modest. So again, the notion of indeterminate potential is key here. So these individuals are also at about a 40% risk for increased risk for all-cause mortality. It was observed in a coupled studies. And that, maybe that doesn't seem surprising because they're at increased risk for blood cancer, but again, blood cancer is pretty rare. It's hard to attribute that all purely to a blood cancer risk. There was important exploratory cause-specific analyses that we did that suggested that maybe it's due to a more common cause of death, like atherosclerotic cardiovascular disease. A few years later, we studied that using a variety of cohorts, exome sequencing cohorts that my mentor, Sate Kathraisin, had ascertained to look at the heritable basis for early onset MI and coronary artery disease. And then we looked for CHIP mutations. We found that individuals who are older who had coronary artery disease with matched controls, including age matched controls and others, and had about twofold enrichment for CHIP mutations. And pretty surprisingly, among individuals who are younger compared to older controls, younger individuals who sustained early onset MI, so in their 40s, had about a fourfold enrichment for CHIP. And the prevalence for CHIP in that group is about 0.5%. But that relationship, prevalence to effect estimate, is similar to familial hypercholesterolemia, well-recognized monogenic heritable factor for early onset coronary artery disease, but readily detectable by lipids. This is, not readily detectable by routine clinical assessments. So those were, nested case control analyses. We subsequently did prospective analyses just to confirm that if you knew that somebody had CHIP or not at baseline, those who had CHIP had greater incident risk for cardiovascular disease. A key discriminator are those who have larger clones. So, going back to our notions of the biology of hematopoietic stem cells and their role in atherosclerosis development, we found that individuals who had CHIP who didn't have cardiovascular disease, they had a greater burden of subclinical atherosclerosis on CT. And again, those with larger clones had more athero. And then we worked with Peter Libby's group at the Brigham to essentially model this in mice, where you would take a mouse, and we started with TET2, you'd knock out TET2 in the mouse, and then take an atherogenic mouse, basically a familial hypercholesterolemia mouse, and then you take the bone marrow of the TET2 deficient mouse, so the hematopoietic stem cells all deficient in TET2, and then transplant that into an atherogenic mouse versus a wild type transplantation. And we find that mice that are deficient in these CHIP genes have a greater burden of atherosclerosis. This is in the aortic root, and then recapitulating the biology that we see in humans. Now, what's helpful for us is to try to understand, is this, despite what we see with the mice, could there be some sort of reverse causation or not, or is this just a marker of aging and it's just generally confounded? It's helpful to understand that there actually are other forms of clonal hematopoiesis that have been described. Another well-characterized form are larger structural variations, meaning chromosomal rearrangements that have been observed in blood cells. These are largely lymphoid phenomena. They have been also linked to blood cancer, largely lymphoid cancers, whereas CHIP's more linked to future risk for AML. They interestingly are also linked to increased risk for mortality, not associated with coronary artery disease or so far other cardiovascular diseases. And recently we found that actually is an important risk factor, age-related risk factor for severe infections, as well as COVID-19. Really honing on the notion that it's maybe not just clonal hematopoiesis, it may be something specific about these mutations. So inflammation has been implicated. I showed that schematic earlier on from Alan Tull talking about some of the key inflammatory mediators of myeloid cells and atherosclerosis in general. But we find, especially in mice with these CHIP mutations, that IL-6, IL-1 beta, these are part of the NLRP3 cascade. These are heightened. CRP is a downstream product. But interestingly, we find that individuals with CHIP only have modest increases in CRP, suggesting that it is capturing a different aspect of inflammation. If you do this more agnostically, this is work by Stephanie Dimmler and Andreas Seiher in Germany. If you just do broad gene expression analyses in individuals with CHIP compared to individuals without, and do an untargeted, unbiased approach, we find basically the same things that light up as far as inflammation and leukocyte activation. So, these things are correlated with the presence of having CHIP. Now, are they key determinants, these inflammatory pathways in going from CHIP to atherosclerosis? Really, the first demonstration of that was a paper simultaneously from our work by Jose Fuster and Ken Walsh when Ken was at BU. Ken's now at VA. And Jose Fuster is Valentin Fuster's nephew. So obviously, bright apple doesn't fall far from the tree. So they took, their mice similarly modeled with CHIP versus not. You can see that's this at baseline. So more atherosclerosis at the aortic root, and then inhibited this pathway. They use an NLRP3 inflammasome inhibitor. And you find, both in the wild type versus knockout, there's been a reduction in atherosclerosis, but a much greater delta among the mice with CHIP. And you can see that here, right here, the larger delta here, again, indicating specificity and more efficiency of actually targeting that pathway in individuals with CHIP. Now, all of you may be thinking, okay, there's another trial that's been done, a phase 3 cardiovascular outcomes trial testing inhibition of this pathway, an IL-1 beta monoclonal antibody, that's canakinumab, and the Cantus trial. These are individuals with atherosclerosis and high CRP. There's about a 15% relative reduction in their primary composite cardiovascular outcome. Now, Novartis, the sponsor of Cantus, motivated by our work, they did targeted sequencing for CHIP genes, looking for CHIP mutations in Cantus. And they found that among the participants of Cantus, those who had TET2 CHIP had a substantially greater benefit from getting canakinumab versus placebo, a hazard ratio of 0.36. Obviously highly exploratory. This is an abstract from a while ago. They've had some leadership changes in their group, but as the paper itself that's working its way through, but quite strikingly different from this. These are effect estimates that we never see in atherosclerosis trials. Now, we were interested, because again, that's reasonably preliminary, but compelling and consistent with the mouse data. Is there a way that we could use our expertise in the germline genetics to test this? Now, it turns out that there is a wonderful therapeutic proxy for this pathway, genetic proxy. There's a common coding mutation in the IL-6 receptor. It's a missense mutation is shown in the general population to be linked to about a 5% reduction in the odds for coronary artery disease. It's been, robust for causal inference for the relevance of this pathway for atherosclerotic cardiovascular disease. And we simply asked, can we recapitulate the efficiency of targeting that pathway among individuals with CHIP? If you take individuals with CHIP and then you compare those who are genetically deficient in signaling through this pathway versus those who aren't, and then you compare it to individuals without CHIP, and then also those differences. Indeed, we find a difference. We'll start on the left panel, individuals without CHIP. And so individuals without CHIP, if they then have that IL-6 receptor mutation, very modest decrease in the incident, in the event rate for incident cardiovascular disease, also about 5%, non-significant, but that could be due to power. There could be still a modest benefit in the general population. And then if you look at individuals with CHIP, there's a much larger difference in the cumulative event rates, different in relative risk and obviously absolute risk. The hazard ratio here is 0.46. So again, now 3 lines of evidence, both 2 in humans and 1 in mice, all different approaches that are converging on the high degree of efficiency of targeting this pathway among individuals with CHIP. And here, actually, if you see this curved CHIP with the IL-6 receptor mutation, almost largely superimposed those without CHIP. So, we've also been interested in trying to understand the root causes of CHIP, towards, primordial prevention, but also better understanding atherosclerosis. This is a paper that came out last year in the TOP-MED consortium. This is an NHLBI effort called Transomics for Precision Medicine to look at all these different features of precision medicine that we've talked about, the multi-omics and the heart, lung, blood, sleep disorders. And we used the whole genome sequencing that was generated in blood cells and did a sort of simultaneous germline somatic genetic analysis. And the reason why we look at the heritable factors, even though we don't think that it's going to play a major role, is that can help us pinpoint to causal factors. So we found, basically generated at that point, the largest collection of individuals with CHIP. There's mainly a technical feat to do it with whole genome sequencing. We find similar gene distributions and prevalences. Now, because we have a larger data set and cohorts that have been fairly well profiled, we can start to look at things by gene. And we do find some interesting differences as far as differences in inflammatory cytokines and others. And I'll come back to some of this. We do find that there are heritable factors that influence the development of CHIP. So this is the gene for telomerase that regulates telomeres. TET2, I keep mentioning that gene is a gene that's implicated in acquired mutations, but actually there's a common germline genetic variant, a low frequency germline genetic variant that's near TET2. Now it turns out that TET2 variant is really exclusively observed in African Americans, and there's been important efforts by NHLBI and TOP-MED to enrich TOP-MED for non-Europeans, actually majority non-European. So among the African Americans, a subset of them have also undergone whole blood transcriptomics, and we find that African Americans who have these mutations have a decreased expression of TET2, in all of their cells. We did some modeling to indicate that the lead variant likely influences an enhancer of TET2. So basically what we observed in the humans with decreased gene expression, we hypothesized because it was disrupting a key regulatory element, and we recapitulated that in cells. This is with Vijay Sankaran, who's the chief of peds hemonc at Boston Children's. And then we find that also, in addition to decreasing TET2 gene expression, we find that that's a necessary determinant in regulation of hematopoietic stem cell colony formation, meaning decreased TET2 also leads to just more cell divisions, and we think providing an important substrate for the acquisition and expansion of CHIP mutations. There's a complex relationship with another one of the loci at the TERT locus. So telomeres have been previously shown to be inversely correlated with a coronary artery disease. If you take individuals with CHIP versus without, correlate with telomere length, those individuals have shortened telomeres. What was then surprising to us was when we did this genetic association analysis, alleles that predispose to telomere lengthening actually lead to increased risk for CHIP. That is actually consistent with the cancer literature as far as lengthening of telomeres leading to increased risk for for solid cancer, really largely. So we did this interesting bidirectional Mendelian randomization analysis where we think the model is telomere lengthening helps predispose to the development of the CHIP mutation, but then the development of CHIP then in the affected cells leads to telomere shortening, and that may be an orthogonal component towards promoting cardiovascular disease. I won't go into too many details, but again, these are epigenetic regulators, the top two genes. When we look in humans, the methylation profiles are actually quite starkly different compared to individuals without. Methylation profiles have been previously linked to cardiovascular disease. And so we're trying to now disentangle to what extent these disruptions influence cardiovascular disease. But some of our colleagues at Stanford, Sid Jaiswal, they have shown that among individuals with CHIP, methylation profiles that have been previously linked to aging and cardiovascular disease are potent discriminators of cardiovascular disease risk, more so among individuals with CHIP versus without. And this is beyond the usual stratification of clonal expansion. So, we've been interested in using this as a tool to try to understand whether CHIP may explain increased risk that has been empirically observed in other subsets where there has been heightened cardiovascular disease risk outsized any relationship with traditional cardiometabolic risk factors. One such group is, women with advanced ovarian aging, premature menopause. We recently observed that premature menopause is linked to a variety of age-related cardiovascular disorders. And in two different cohorts, we find that individuals who sustained, who had premature menopause have a greater prevalence of CHIP mutations, also actually other forms of clonality as well too. And that's particularly so for the women who undergo premature natural menopause and not surgical menopause. That in turn is obviously linked to heightened risk for cardiovascular disease. HIV has been, appreciated for the last few decades as the epidemiology has shifted and the age of individuals with HIV has also shifted to the right. HIV is an important risk factor for cardiovascular disease. It's linked to a variety of cardiometabolic alterations, but still the risk is outsized those. We found recently that individuals with HIV in an exploratory analysis had stark increases in the prevalence of CHIP mutations, especially as they got to middle age. And that was really nicely shown in a much larger cohort, also demonstrating basically these exact same relationships. And we have a lot of efforts now to look at this in the context of a variety of HIV cohorts. Also, diet obviously has been shown to influence cardiovascular disease risk independent of cardiovascular disease risk factors. Diet is important also for cancer. We use the UK Biobank and tabulated a variety of dietary scores to capture healthy and unhealthy diets, and we find that individuals who have unhealthy diets independent of all the other risk factors are enriched for CHIP mutations. And again, diet also represents an important stratification for CHIP-associated cardiovascular disease, lending to the hypothesis that maybe improved diets may reduce the risk of developing CHIP and may also reduce the risk of CHIP-associated cardiovascular disease as a tool that's already available. Now, what about other conditions, other cardiovascular conditions? A couple studies have looked at the implications of CHIP among individuals who already have cardiovascular disease, namely both heart failure and aortic stenosis, a population that gone on to go get TAVR. But if you look at these patients who have CHIP mutations versus not, those who have CHIP mutations have starkly worse prognosis as it relates to all-cause mortality. That's now been appreciated in a number of different studies. And in those populations, the presence of CHIP mutations, mortality is similar to the short-term mortalities that you see in cancer itself. And they're not dying of cancers. There's been additional work to look at CHIP as it relates to heart failure. This is work also led by Ken Walsh, looking at both ischemic and non-ischemic models of heart failure in mice and showing that CHIP promotes the adverse remodeling and some surrogates of heart failure in these mice. And recently we used the some of the cohorts in TopMed and the UK Biobank to show that CHIP also is a contributor to the development of heart failure, both in individuals who have ischemic etiologies for their heart failure, and then individuals who have no known coronary artery disease who also develop heart failure. So, we think that CHIP represents a new risk factor for coronary artery disease, not readily identifiable by any current clinical assessments. So CRP is relatively similar, and then the CBCs are normal, lipids and blood pressure are all normal. There's a particular axis that we've honed in on that we think might be effective at reducing CAD risk conferred by CHIP. And we've basically been focusing on the same 5 risk factors that show high degree of efficiency in treating those pathways when they're altered. When we think about other things like polygenic risk scores and others, those are largely still allocations of medicines that treat those other targetable, those other pathways as well recognized pathways. This represents a unique approach where we find a new risk factor, and then we use potentially medicines that we would not consider in other contexts to reduce risk for a common disease. We continue to look at a variety of risk factors for the development of CHIP to help us understand new insights into what promotes cardiovascular disease, and to try to best understand who might benefit from screening for these kinds of tests and detecting CHIP. And importantly, all of these studies all represent so far all cross-sectional analyses. This is all a very new field. It's really like a 5-year-old field. But there's been a fair amount of investments from sponsors. Lots of people are interested. For many people, especially if they have whole exome sequencing in their cohorts, it's largely just a computational exercise to get this information and to start to learn more. But we're now really interested in looking at the natural history, the longitudinal factors. If you actually sequence older individuals deeply, almost everybody has pathogenic leukemia mutations that are largely quiescent in middle age. And so there are some necessary set of factors that are required with clonal expansion. So if you identify these things early in individuals, how can you interrupt that process? And that I think will continue to gain insights as we do more longitudinal studies. So for the work that I described here, lots of collaborators, local and beyond. These are humongous efforts that involve tons and tons of people. So this is just a small snapshot. And this is a group in the lab that helps, that really makes all of this work happen. And just a handful of key folks that are either currently or formerly linked to Yale. So Miriam is currently an MD PhD student in her last year at Yale. Probably recognize Katie Clapham, who's in the cardiology division there, who's now at the Brigham, but doing her postdoctoral work with us. And then Aaron Small and Sarah Urbit, who were recently residents at Yale and are now in the lab. So great, thank you. I think time for questions.
Thanks, Pradeep. That was an excellent talk. So please go ahead and raise your hands or just ask a question right away. But if you feel better about writing a question in the messages, I can read them out. Arya has a question there.
Phenomenal work, Pradeep. My question to you is that obviously these CHIP mutations are very few cancer-causing mutations, and obviously they are epigenetic regulator, therefore they have a high impact. But how much of the aging you think is related diseases caused by this coronal expansion, the contribution of that, how much you guess? And the other thing is that you didn't mention about the effect of exercise on, reducing this kind of CHIP expansion, the expansion of coronal expansion. If you can have your insight, that would be great.
Yeah, no, I think this is great insight. So as we talked about in the beginning, age is a very powerful risk factor for age-related diseases, right? To what extent does CHIP play? Because CHIP is not very common. This is only, this does not fully explain age and its relationship to cardiovascular disease or other conditions. It is a contributor. I think I'm, at least scientifically quite curious about other tissues, CHIP has been a story of convenience because we do sequencing on blood cells. So I think there may be other examples, and there are increasingly so, of somatic mutations and clonality in other tissues that influence other diseases, but also including cardiovascular disease. There's great work from Irv Weissman and Nick Leeper in Stanford looking at clonality in endothelial cells, a separate phenomenon from CHIP, also promoting atherosclerosis. So I think there may be other genomic models of aging that may influence age-related cardiovascular disease, but here is a very tight molecularly defined subset of which age happens to be a risk factor, but there are certainly maybe others. And then remind me what the other question was.
The exercise effect on this.
Oh yeah, exercise. We're starting to look at that. I would say it's not been so compelling yet. But again, there's great biology, from Matthias Nierendorf here and Phil Swirsky on sleep and clonal hematopoiesis. So we're starting to look at sleep. Exercise may play a role. Again, a large part of it is how is this captured? How well is this captured? And can we study this well? So we're starting to look at these things. Undoubtedly, the things that influence the development of CHIP and expansion are not heritable factors. They relate to lifestyle and environmental factors.
Perfect. Next up, I think we have Erica Spatz.
Hi, Pradeep. This is a great talk, and I'm a big fan of your work.
Thank you.
I was curious to get thoughts on as you mentioned, with Cantus, with canakinumab, with inclisiran or colchicine, or diets and exercise. How do you see this playing out in the future in terms of better phenotyping people's individual residual risk, and then more, applying more tailored therapies that may target the actual mechanisms of that increased risk? Right now, it feels very overwhelming to implement a lot of the newer medications in that risk model without further phenotyping who might benefit most.
Yeah, I think, Erica, you're hitting on really important things, that there still needs to be more work done as far as what are the right therapies, but also better trying to identify, maybe even further risk stratify who would benefit the most from therapies. I think that's where the natural history is going to be most helpful, because it's also, it still is indeterminate potential as it relates to coronary artery disease. It's not like everybody's getting coronary artery disease. And some of these risk discriminating factors, like how large is the clone, meaning what fraction of the blood cells are implicated in the mutation. Now, great work from Sid at Stanford that suggests methylation profiles may be helpful. It was interesting when I was talking to a reporter once who was interested in deciding whether they wanted to write a story on CHIP and talking about this IL-6 receptor work. One thing that I hadn't thought about that she pointed out was like, oh, if you were naturally deficient in IL-6 signaling and you had a CHIP mutation, maybe you actually don't even need inhibitor for that pathway. That may be actually an important discriminator as well too. So I think the longitudinal analyses and then additional profiling, additional phenotyping to better look at the discriminators. And then, as I highlighted, there may be other orthogonal pathways, and some of it may be different from gene to gene. But that we're only understanding as the data sets get larger, because it's not like everybody has CHIP. We need much larger data sets to understand that. And then that last point of looking in other clinical trials, because as you point out, inflammation and this pathway, including the NLRP3 inflammasome, has been implicated in other therapies, including colchicine that's already widely available. I know this is on the mind of a lot of the people who run the colchicine trials. A major, I'm not a trialist, but a major challenge is, initially making sure there is a plan to doing the genetic analyses when planning the trials. In my experience, trying to come in afterwards, if people have not thought about it, not have budgeted for it, very hard to execute. Some of the trialist groups are very good at making sure that that is part of the plan early on. But I think, especially with the excitement from germline genetics and now this, I think sponsors are now, it's a relatively modest addition to their budget that they'll start looking. The nice thing, you also mentioned inclisiran, and is there a role for other cholesterol lowering strategies? At least this is kind of on the radar now, I think, for many larger pharma companies, that there is now investment, and we're hoping to look at things like this in inclisiran and others.
Great.
I think it's Dr. Jeff Bender.
Pradeep, that was both a wonderful talk and obviously paradigm-changing work. It's remarkable, remarkable work. So as a little bit of an extension to Erica's therapeutic targeting question, obviously the NLRP3 IL-1 beta pathway is linked to CHIP. And one of the concerns of chronic suppression, if you will, of any immune and inflammatory pathway is chronic immunosuppression, which would
Right.
be unfavorable. So in your crystal ball of the future, is there any thought about the ability to selectively target the CHIP clone, the expanded CHIP clone in an inflammatory pathway to really hone in on the bad player and really remove the risk? I don't know whether that would do it, but it's just a thought.
Yeah, no, definitely there are notions, at least in blood cancer and blood cancer targeting, of developing specific, immunotherapies and other targeted therapies for mutations, or at least linked directly to mutations that are implicated for specific cells to mitigate sort of off-target stuff. And I don't know how many people are thinking about, or at least trying to do that as it relates to cardiovascular disease, but I think people are also just generally thinking about how do you prevent the progression of CHIP, because we're finding increasingly so, obviously other age-related cardiovascular disease, but there are other non-onc, non-cardiovascular conditions that are implicated. Good news is that it's not everything related to aging. If it was everything, then I would say, oh, then we just have a good biomarker for age. But it may be, I think, as you highlight, Jeff, effective to do it earlier on. There are some targets that we've developed based on the germline genetics to look at what are the heritable factors that promote the expansion of CHIP clones. There's a lot of more basic work being done by one of my former postdocs, Alex Bick, who's at Vanderbilt, also in collaboration with Sid at Stanford, to really figure out if that is a viable therapeutic target. Like if you find these clones earlier on, is there a way to mitigate, but then also make it more specific to the CHIP cells? But there are, I think, other inhibitors in this pathway that are, I think, better than sort of, as you know, in Cantus, the major adverse effect was sepsis among patients with canakinumab. You could say that risk-benefit ratio for canakinumab itself may be better in individuals with CHIP, but ideally you'd have a lower risk therapeutic. Ziltivekimab that was acquired by Novo Nordisk from Corvidia is a very specific IL-6 inhibitor that seems to be more potent in its CRP reductions. In their phase 2 programs so far, they've not seen a link to infection, and they think that sort of specificity of IL-6 targeting might be better. But the people, like Peter Libby, Alan Tall, all those folks, they think a lot about what is the best way to target that pathway to mitigate some of these immunosuppressive effects. So I think there's lots of thought and thinking about that. But then I think there's other strategies to being more specific to the cells that, great hypotheses and stuff out there. There is some work that's happening, but early.
Thanks, everyone. Any other questions? So
go
ahead, John.
I mean, it was a wonderful presentation. I suppose the focus has really very much been on the negative aspects of CHIP. And given the sort of that almost all centurions have CHIP, could there be any evolutionary benefits of CHIP?
It's an interesting question. There are individuals who are in their 80s and 90s that almost all of their blood cells are a reflection of clonal hematopoiesis, singular clone with these CHIP mutations. And, hematopoiesis is largely a polyclonal event. Here, we're just talking about specific selection of a single clone. Some of the challenges are, I think, a selective bias when looking at really advanced age individuals, because there is, potentially some other protective factors that have allowed them to be to that age. So one can use that as an opportunity to look at what are the factors, what are the resilience factors in the setting of clonal hematopoiesis. But at least I'm not aware that anyone has looked at whether fitness of clonally expanded mutations has some advantage. I'm sure there is some aspect of it as advantageous. I mean, at least in our society, where the age of the modern human is much greater than others, we largely see the downstream consequences of maybe persistent clonality. But maybe there is some short-term benefit. It's interesting thought, interesting question. I mean, people also see this for Lp(a), right? You're talking about atherosclerotic cardiovascular disease linked to only bad things, but if you look closely, it has a very interesting relationship where the risk goes down a little bit and then goes up. And people who think about this a lot, like Sam Semikis, they think that it has some prior benefit as it relates to infection, but obviously when it's too high, has problems. Could be something like that for CHIP. I don't know.
Great, thanks.
Perfect. Thanks. And Pradeep, do you often think about these, not just the mechanistic aspect of things, but also about how to apply them to populations, just as your role as a clinician? So how do you sort of, see the implementation gap being bridged here for, a lot of insights that, you derive and others have derived in this area?
Yeah, that's a great question. So I think there's lots of excitement as a risk factor, unrecognized by current clinical assessments, and then maybe therapeutics, maybe things that are even available like colchicine, we don't know. But the question is, how do you start identifying? And we get some of these referrals today. In general, though, it's not bona fide CHIP. It's largely people who have undergone sequencing, these so-called blood biopsies, which include all of the CHIP mutations, largely for some CBC abnormality. So there's a phenomenon called CCUS, clonal cytopenia of unknown significance. That's CHIP basically with cytopenias. So individuals out there, very low risk for malignant transformation, and the hematologists are attuned that actually atherosclerotic cardiovascular disease may be a greater absolute risk increase. So we get those folks referred, and sometimes low-grade MDS. Very rare that somebody is known to have CHIP unless they participate in a research study, or at Memorial Sloan Kettering, where part of protocol, basically everybody who walks in the door gets testing. And so they have a lot of people largely with other cancers who also have CHIP mutations. Large evidence gap on what to do as far as prospective studies, even though there's good observational stuff and post-hoc RCT stuff. But I think the important, so still a lot of, I think, research to be done, including prospectively. But the assays for detecting CHIP mutations could be quite efficient. Meaning, one does not have to do whole exome sequencing. Really, the top 10 genes is where you get, at least 90–95% of CHIP. And targeted sequencing of those top 10 genes actually only costs about like $20. Some of these assays are out there, not necessarily with some of the other factors that discriminate from CHIP, but we've been working on an assay so that people can do this. And that has actually enabled a variety of cohorts that have come and collaborated to now more efficiently do CHIP testing at larger scale. But I think those are the things that cost effectiveness for the assay that'll be necessary to get it into the therapeutic space. So I think that aspect, looking at the longitudinal history, because like when somebody gets the assay, if you sequence deeply enough, people will have smaller clones. How do you follow them? And then I think as we have more and more healthcare associated biobanks, the prospect of molecularly driven clinical trials is increasingly feasible. And we are actually working with some folks to design those kinds of studies as it relates to CHIP. So I'm pretty excited and optimistic to be kind of some of the first demonstrations as it relates to atherosclerotic cardiovascular disease. So we're getting there, but I think there's still more work to be done.
Oh, thank you. Thanks a lot. It was a great presentation and I think a great Q&A. So if we don't have any other questions, we might end a couple of minutes early. Thanks again, Pradeep, for visiting us, and hopefully we get to see you in person soon.
Yeah, of course. Great questions. Thanks, everyone.
Bye.
Have a good day.